Hello, I'm Professor Brown, and we're going to review how to do a bacterial smear, after which we're going to do a gram stain. So what we need to start with is a nice clean slide. So what I like to do is to wash the slide first with soap and water over at the sink, and I've already done that, and then dry it with a paper towel, and then you're ready to go and you have a nice clean surface to create your bacterial smear. Now, a bacterial smear can be made in two ways. It depends on what kind of culture you're getting your bacterial from, a liquid one or a broth, or a bacterial culture that's on solid medium, which we call either an agar slant or an agar plate. What we're going to do is we're going to create a smear from an agar slant. So the whole point is you need a little bit of liquid on the slide surface. If you're using a broth, all you need is to get a little bit of your broth culture in an inoculating loop and transfer it to the slide and then spread it around. What I'm going to do first, though, is we need a little liquid before we get the bacterial culture from the agar slant. So this liquid doesn't have to be sterile or anything special, just plain water. So my water bottle, I'll get a loop full. You just stick your inoculating loop in the water and transfer it to the top of the slide, right where you want to make the smear. So now we have a little liquid we can spread the bacteria in. And what I'm going to do next, now we're going to do an aseptic transfer of bacteria onto the slide surface. So I'm going to start with this bacterial species right here, is Staphylococcus epidermidis. Now I'm going to do something a little bit unusual. I'm going to make a single smear that has two bacterial species, just for the sake of showing a gram stain. Normally you only have one bacterial species, but you can also do two, and I'll demonstrate that in a moment. So first of all, you need to sterilize your inoculating loop. So you're going to place it always at an angle in the flame, right above the inner cone, starting at the base, waiting for it to turn orange, and then moving towards the tip. And now everything on that inoculating loop has been incinerated and it's now sterile. It's a little bit hot, but I'll show you how we can cool that down in just a moment. Now, whenever we open our culture tube, we want to give it a brief flame in the mouth of the tube. And then without setting the cap down, hold it at an angle like this. And then I'm going to cool down the loop by just touching the agar off to the side. And then gently just touching to get a tiny bit of bacteria, the smallest visible amount on the loop. And then flame the tube again, replace the cap, and we'll set that culture aside. Now I'm going to transfer just a little bit of the bacteria and get it into our little droplet of water. So I'm going to turn it over on both sides. Now we're not going to spread it out just yet because we want a little bit of we want the liquid on there when we transfer the next sample of bacteria. So I'm going to, of course, flame the loop again from the base towards the tip. This is very important because you don't want to create any microscopic aerosols that might contain live bacteria. If you heat the tip or the loop directly, you might get that happening. By flaming from the base to the tip, you get a little more gradual heating, and that will help avoid that problem. Okay, so I've sterilized the inoculating loop, and we're ready for a little bit of Escherichia coli. So we're going to add that to our smear in the same fashion. Again, this is probably a little hot. I'm going to touch the tip of the loop on the agar and the slant first, and then get a tiny bit of bacteria on the end of the loop there, just the smallest visible amount. Okay, set this back in the rack. And now in that remaining liquid, I'm going to spread some of the E. coli. You can probably see it get a little cloudy there. And now that all the bacteria we need are in there, I'm going to kind of spread out the bacteria in this liquid as best we can. By doing this, you can kind of draw a circle or you can go side to side. The idea is to, number one, spread the bacteria as much as you can over the surface of the slide. And number two, you're also spreading out the liquid so it'll dry out faster for you. Okay, and always after you're finished, sterilize your loop. Again, from base to tip. And to complete the smear, we have to fix it. We're going to heat fix it. Now, it's air dried right now, but the bacteria are not fixed to the surface. So I'm going to need to heat fix it. So I'm going to take the slide, and I'm going to be holding it with this fancy slide holder, okay, clothespin. And in our flame, just put it in the flame, moving it around for about 2-3 seconds. 1, 2, 3, right in there over the inner cone. So you heat up the slide, but not too much. But that's plenty of heating to heat fix the bacteria to the slide surface. So having heated them, they'll now be fixed and they won't come off when you do the staining. So now that we have a heat fixed bacterial smear, we're ready to start staining. So I'm going to place my slide on the staining rack over the staining tray. And this is the point where you're probably going to want to put some gloves on, because the stain can get on your fingers. Not that it's that really toxic or anything, but it discolors your fingers, and nobody wants to go home with purple fingers. So, and it's also a good idea to wear a lab coat. So if any stain gets spilled, again, you're protecting your clothes as well. Okay, so now I'm ready to stain. And we're going to do a gram stain first. And we're going to stain this slide doing a gram stain. So the first ingredients, or the first step in doing a gram stain, is to cover the slide or cover the smear with the stain Crystal Violet. This is our primary stain. So add enough to completely cover the smear and leave it for 1 full minute. So now that we've left the Crystal Violet on the smear for a full minute, we're ready to go to the next step. Before we do that though, we're going to have to rinse off the Crystal Violet with water. So with my gloved hand, just take the edge of the slide and then just rinse well with water, holding it at an angle. And if you need to, rinse the back to get any excess stain off. And now we're ready for the next step, which is to add some grams iodine. So the grams iodine, this is what we call a mordant. So we're going to cover the smear with the grams iodine for a full minute. This is a mordant. It doesn't actually stain, but it enhances the primary stain. It enhances the Crystal Violet, so it will stain in the way we want to. So now that it's been a minute, we will take our water bottle and rinse off the grams iodine solution. Okay, and make sure it's all off. Now at this point, all the bacteria on the slide, whether they're going to stain gram negative or gram positive, they're all purple. They're all stained with the Crystal Violet. What's going to happen next is we're going to use the decolorizer. So this decolorizer is a 50% alcohol, 50% acetone solution. This is going to remove the Crystal Violet from the gram negative cells without removing it from the cell walls of the gram positive cells. To make sure it unfolds as it's supposed to, or decolorizes as it's supposed to, you don't want to overdo it. That's why this is such an important step. Your instructions probably tell you to decolorize until the runoff appears clear. For many students, that can be 30, 40 seconds, when in reality it's supposed to be 2 or 3 seconds. So about 2 or 3 seconds is plenty. So I'm going to, again, tilt the slide a little bit and take the gram decolorizer and let it run over the slide surface for about 2 or 3 seconds, and that's plenty. So what just happened, and now rinse with water, what just happened, you probably saw a little bit of purple coming off there. The Crystal Violet that was in the cell walls of the gram negative cells was just removed by this decolorization step. Whereas the gram positive cells, the Crystal Violet was retained, basically, due to the nature of their cell walls. So now we have a smear in which the gram positive cells are purple, and now the gram negative cells are going to be unstained. They're going to be non-colored. So we're going to finish off the procedure, the last step, step number 4, by doing a counter stain with the stain called safranin, which will stain cells pink. So I'm going to cover the smear with safranin for a full minute. All right, now that it's been a full minute, we're again going to rinse with water to get all the excess safranin off the slide. And this step is important because it's going to allow you to see the gram negative cells. The gram negative cells, which were unstained after the decolorization step, the gram negative cells are now going to be stained pink, whereas the gram positive cells will be purple. All right? So now that we're finished with the staining procedure, we're going to remove the excess liquid. So I'm going to take a little bibulous paper here and gently place the slide and pressing very gently just to get all the excess liquid off. Don't press too hard or you might break the slide. And then just let it air dry for a couple of minutes to get any excess liquid off there. And then you're ready to look at it under the microscope. Okay, so now that we're done staining for the day, we need to dispose of the staining waste properly. In the past, you actually could just put it down the drain, but that's now illegal, and we will get fined if we're ever caught putting staining waste down the drain. So by staining waste, I'm referring to all of this material in our staining tray. So again, do not, whatever you do not dump this down the sink. You need to dispose of it in the jugs that are actually in the sink, but they're special jugs with funnels on top. Just dump it all in there, rinse with a little bit of water, and put the rinse in there, and then you're good to go.